Molecular basis of inheritance. What is the meaning of a molecular basis of inheritance? Inheritance means transmission of characters from parents to offspring. How the characters are transmitted means through a molecule. The molecule means either DNA or RNA. Hence, the topic name molecular basis of inheritance. Now, we shall study the nucleic acids. The nucleic acids are polymer of nucleotides. That is the definition for nucleic acid. The nucleic acids are very big molecules or macromolecules present in all living cell, that is from bacteria to higher plants and animals. Now, types of nucleic acids. Coming to the types of nucleic acids found in living organisms are two types. First one, deoxyribonucleic acid, in brief DNA, and ribonucleic acid, in brief RNA. The DNA acts as genetic material in majority of living organisms. RNA also acts as genetic material in some virus. For example, TME virus, human HIV virus. Now, we shall study the discovery of nucleic acids. The nucleic acids were discovered by F. Mischer in the year 1869 from pus cells of hospital bandages. He coined the term nuclein, but later on another man, Altman, coined the term nucleic acids. Why he called nucleic acid means these nucleic acids contain acid. Acid means the phosphoric acid present in the form of phosphate, hence nucleic acids. Now we shall study the DNA. What is DNA? Definition. DNA is a long polymer of deoxyribonucleotides. As I already told you, it acts as the genetic material of living or majority of living organisms. This carries the coded information from one generation to another generation. Okay. Now we shall see the length of DNA. The length of DNA always depends upon the number of nucleotides or base pairs present in it. Say, for example, the bacteriophage 174 contain 5,386 nucleotides. Lambda phase contains 48,502 nucleotides. The E. coli bacterium contains 4.6 into 10 to the power of 6 base pairs. The human haploid cell contains 3.3 into 10 to the power of 9 base pairs. It is the characteristic of an organism also. Now we shall study the structure of in eukaryotic cell. The DNA is found in the nucleus. It is the chief component of chromosomes. DNA also found in the mitochondria and chloroplast, that is, in some cell organelles, DNA is present. Chemically, the DNA is made up of deoxyribose sugar, phosphate group, and nitrogenous base. So you can say the nucleotide. What is the definition of nucleotide? Nucleotide is the fundamental unit of DNA or RNA made up of 3 units, namely nitrogenous base, sugar, and phosphate. Now we shall see the deoxyribose sugar. Deoxyribose sugar present in the DNA molecule. It is a pentose sugar having the molecular formula C5H10O4. You can see the structural formula also. Now, what is very important is in this sugar, one of the oxygen atom or the molecule is absent at the second carbon position. Hence, it is called deoxyribose sugar. If we examine the ribose sugar, OH is present at the second carbon position. Hence, the sugar of RNA is called ribose sugar. Now, we shall study the phosphate group. The phosphoric acid is present in the DNA. The phosphoric acid is present in the form of phosphate group. It is similar both in DNA and RNA. Its molecular formula is H3PO4. This is responsible for acidic nature of DNA or nucleic acids. Why we call nucleic acid means because of the presence of an acid, phosphoric acid. So now nitrogenous base. The nitrogenous base means nitrogen containing compounds. Nitrogenous bases are present in DNA and RNA also. The nitrogenous bases are of two types, are classified into two types. They are one, purines, and second, pyrimidines. Now we shall study the purines and pyrimidines. First, purines. What are purines? How to define purines? Purines are the double ring heterocyclic compounds. Double ring means having a benzene ring and an amide group. Thus, two rings are there. The purines are two types, namely the adenine and guanine. The purine present in DNA are adenine and guanine. Adenine in brief A, guanine in brief G. You can see the structural formula of both adenine and guanine. Now we shall study the pyrimidine. Pyrimidines, what are pyrimidines? Pyrimidines are the single ring structural compounds. Single ring means only the benzene ring is present. The pyrimidines are two types. That is the pyrimidine of DNA, cytosine and thymine. Cytosine in brief C, thymine in brief T. But another uracil pyrimidine is present in RNA. So this is the difference between the DNA and RNA. That is, in DNA, thymine is present. But in RNA, instead of thymine, uracil is present. Now you can see the common pyrimidine for purine and pyrimidine. The common one is cytosine. Cytosine is common in DNA and RNA. So that's all about the pyrimidine. You can see the structure also in the diagram. Now we shall study the nucleosides. What is the meaning of nucleoside? Nucleosides are compounds formed by the combination of pentose sugar and a nitrogenous base. Or you can say a nucleotide without phosphate is called nucleoside. nucleoside. Nucleoside is different from nucleotide. Now, how the nucleoside is formed means a nitrogen base attached to the C1, that is carbon 1, position of pentose sugar by the formation of a special type of bond or linkage called N-glycosidic bond. Very important for 1 mark. In DNA, the nucleotides are formed by the combination of deoxyribose sugar and nitrogenous base. Now, we shall study the types of nucleosides of DNA. As I told you, the base and the sugar forms nucleoside. Adenine base and deoxyribose sugar form deoxyadenosine. Guanine and deoxyribose sugar forms deoxyguanosine. Cytosine and deoxyribose sugar forms deoxycytidine. Thymine and deoxyribose sugar forms deoxythymidine. So since 4 types of bases are there, 4 types of nucleosides are formed in DNA. Once again, they are deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. It is for 2 marks, half mark each one. Now, the nucleotides. I already defined the nucleotide. The nucleotide always formed by the combination of phosphate and base and phosphate, base and sugar. Or you can say phosphate group when linked with nucleosides forms nucleotides. The nucleotides of DNA. Since 4 nucleosides are there in DNA, 4 nucleotides are formed. The 4 nucleotides of DNA are 1. Deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate. In brief, deoxyadenosine monophosphate called DAMP. Likewise, DGMP, DCMP and DTMP. It is for 2 marks. Now, for example, write the components of deoxyadenosine monophosphate. Question is for 3 marks. So you have to write the base, adenine. You have to write the sugar, deoxyribose sugar, and the phosphate. 3 units carries 3 marks. So that's all about the bases, nucleotides and nucleosides. Now we shall study the structure of polynucleotide chain. What is polynucleotide chain? That's very important. Definition. A polynucleotide chain is formed by the addition of at least 5 nucleotides. 5 nucleotides. A polynucleotide chain is one which consists of more than 5 nucleotides. These nucleotides are joined end to end and form a big molecule or polynucleotide chain. The number of nucleotides linked each other, or you can say two nucleotides are linked each other by the formation of another bond called phosphodiester bond. So thus a long chain of polynucleotide strand is formed. Now, the phosphodiester bond forms between the fifth and the third carbon atom position of a pentose sugar. This strand we call 5' and 3' strand. Therefore, polynucleotide strand contains 5' and 3' end. In DNA, other strand is 3' and 5'. Now, we shall study the structure of DNA. DNA double helix, or write the silent features of DNA. It's very important for 5 marks. It was in the year 1953. J.D. Watson and F.C. Crick first proposed a model in order to explain the structure of DNA. Now it is popularly known as Watson and Crick model of DNA. The double helical model of DNA was based on the work of X-ray diffraction studies made by M. Wilkins and Rosalind Franklin. Later on, it was in the year 1962, they got or shared Nobel Prize. They shared Nobel Prize along with M. Wilkins. Now we shall see the structure of double helix. The characteristic features or silent features of helix or DNA. According to Watson and Crick, model of DNA. The DNA is made up of two polynucleotide chains or strands. These two strands formed by phosphate and sugar. The phosphate and sugar forms the backbone for the helix. The bases project inside the helix. Next point, the two polynucleotide chains run in anti-parallel direction, one in 5' and 3' and other in 5' and 3' and 5' direction. That is, we can say it got polarity. Next point. The two polynucleotide chain is made up of only nucleotides. The nucleotides are linked by the formation of phosphodiester bonds. Next point. The two chains are coiled in right-handed manner. ನಾವು ಹಗ್ಗ ಮಾಡಬೇಕಾದ್ರೆ ಬಲಗಡೆಗೆ ಉರಿ ಹಾಕೋದು ಅಂತ ಹೇಳ್ತೀವಿ. ಆ ಥರ right-handed manner and forms the right-handed helix. Such type of DNA is called B-DNA. It is present in living organisms. The other DNA is also there, that is forms of DNA. For example, cDNA, zDNA, aDNA, etc. The two polynucleotide chains are held together by the formation of hydrogen bonds. Between purine and pyrimidine bases. The pairing of bases is specific or particular. Adenine always pairs with thymine by double hydrogen bond. Guanine always paired with cytosine by 3 hydrogen bonds. So because of this reason, the thickness of DNA remains constant. This pairing of bases or specific pairing of bases was discovered by E. Chergaff. It's also called base equivalence rule of Erwin Chergaff. According to this, the amount of purine equals to the amount of pyrimidine. A plus G equals T plus C. The amount of adenine equal to the thymine and the amount of guanine equal to cytosine. That is A equals T and G equals C. The ratio of A plus T over G plus C always remain constant. Another feature, it is, as I already told you, a right-handed helix and take one complete turn at every 34 angstroms, which contain, or one turn contain, 10 nucleotides. So the distance between two bases is 3.4 angstroms. Another character. The two chains are complementary to each other. Complementary means if you know the structure of one strand, you can easily write the another strand. That is what we call complementary to each other. Then the diameter or thickness of the strand is 20 angstroms. 20 angstroms. Another feature, it shows major and minor grooves. So these are some of the characteristics of Watson and Crick DNA. Okay, now see the diagrams of double helix. It is right-handed helix. You can see the complete turn, that is 10 nucleotides in each turn. Now, after the discovery of structure of DNA, helix, F.H.C. Crick proposed the central dogma of biology. The central dogma of biology states, or molecular biology states, that the genetic information always flows from DNA to RNA, from RNA to protein. That is, from the information present in DNA is passed to RNA. This process we call transcription. From RNA, a protein is manufactured. This is called translation. That's central dogma for 2 marks. You may write, or you can see the sequential thing, DNA to RNA, RNA to protein. But later on, in retrovirus, Temin discovered reverse transcription. That is, genetic information flows from RNA to DNA, not from DNA to RNA. This phenomenon called Taminism, for which got Nobel Prize.